So let me first of all introduce Colin Collins. And so Colin is a senior scientist at the Vancouver Prostate Centre and a director of the microarray facility, an associate adjunct professor at the University of California, San Francisco, Helen Diller Family Comprehensive Cancer Center, and a visiting scientist at Lawrence Berkeley National Lab. His current research is best described as translational genomics, where mathematics, genomics, computer science, and clinical science converge in diagnostic therapeutics. And he has a long history of translating this into really interesting results. And I'll let Colin go from here. Please welcome Colin.
Well, thank you. It's really a tremendous honor to be here. I thank Genome and GBC for their support over the last 2 years. So I think I have a long history of trying to translate genomics. Hopefully now we're going to do it in a city with such incredible infrastructure. So yeah, what I'm going to talk about is translating genomics for personalized therapies in prostate cancer. This has always been my last slide. I moved it up to my first slide. So this is the problem in prostate cancer. Live long, prosper, and increase your risk of prostate cancer, or there's no free lunch. 1 in 55 men between the ages of 40 and 59 will develop invasive prostate cancer. This increases to 1 in 7 for men between 60 and 70. The median age of diagnosis is 66, and organ-confined tumors don't typically affect survival for 10 to 15 years. So it's critical that patients be identified who will metastasize and die of disease within their lifetime. And this becomes even more critical as lifespans increase. So we really need biomarkers to stratify men into low-risk and high-risk groups. This is made all the more urgent by PSA. This is from a good friend of mine at UCSF, Matt Cooperberg, who's shown the effect of PSA screening over time on prostate cancer diagnosis. So we see that in the early 90s, about a quarter of guys are coming in with real low-risk disease, and half of guys are coming in with advanced stage, usually metastatic, life-threatening disease. And over time, this is flipped. So now we have 25% of guys are showing up in real trouble, and about 50% of the guys who are showing up don't really know what to do about it. It's very low-risk disease. It's not clear they're ever going to die of this disease. And in this vexing problem in the middle, this intermediate group, roughly half these guys will go on to progress, half won't, and so we really don't know what to do. And so the cost of this screening, this sort of inversion, is really a significant overtreatment. Lots of guys are being treated who don't necessarily need to be. And so that results in this. So we have about, let's say North America, we have about 250,000 diagnoses a year, 30,000 deaths. This results in about roughly 200,000 radical prostatectomies and 100,000 radiotherapies. Both of these are associated with significant morbidities. And 30 to 50% might be avoided. So the guys going what's called active surveillance. And then in North America, that could be worth $2 billion just for the healthcare system, just getting guys to, stay healthy and whole. So we asked a couple years ago, which is more than that now, if we could use genomics to identify men who would be candidates for active surveillance, so sort of non-therapy. We watch very carefully, but don't have an intervention. And we developed a group of genome-based biomarkers called GEMCAP, and we showed that in fact we can stratify candidates for active surveillance and candidates for aggressive intervention in a small study. In a larger study, but still a small study, we found, we just published in 2010, we validated that smaller study, and we found that the GEMCAP signatures were able to detect the cases with negative lymph nodes recurred within 1 year of surgery more often than this Catan nomogram, which is really a clinical sort of gold standard for predicting disease-free recurrence. And we clearly detected additional benefit of the GEMCAP genomic signature in predicting disease progression. So that made us happy. But then Mike Catan, who developed the nomogram, noted we needed to do a larger set of tumors to be able to get more data. And Charles Sawyer's group then published a paper where they looked at 240 so tumors, and they confirmed our finding, but were able to substratify. So they can show different groups. So the guys up here would be good candidates for active surveillance. These guys are in trouble, need an aggressive intervention. So that was really nice. So I think that the work going on in Vancouver, Eli, and work going on in California, and Memorial, other places, these biomarkers are going to come online. So what I'm interested in doing now is sequencing progressive prostate cancer to identify therapeutic targets. And in prostate cancer, this molecule, the androgen receptor, really defines prostate cancer, sort of the central axis of the disease. And then this molecule will become amplified, mutated, or undergo alternative splicing to defeat any therapy that's thrown at it. And so we want to understand how it does that. And this is a collaboration with Amina Zabidi and Melanie Lehman and Pfizer. And what we're asking is, can we identify the molecular mechanism of resistance to therapy? So what we do is we put human tumors, in this case cell line, into mice as a xenograft, castrate, the tumor dies or shrinks. We then apply second generation androgens here, and then we either get good response, no response, or intermediate responses. And then we can use what's called a correspondence analysis, which groups these together. So again, these are the ones who are sensitive to the drug, these are resistant to the drug, and these are the initial state. And we're picking some of these and sequencing transcriptomes and genomes to try to identify the molecular determinants of resistance to these second generation anti-androgens. Another way to approach this is we know that the androgen receptor undergoes alternative splicing as a mechanism to defeat molecules that bind certain regions of the androgen receptor. And so Anna Lipokimada, who moved up from Lawrence Berkeley, has developed these algorithms for predicting alternative splicing, microarray data, and RNA-seq data. And this just shows that she's able to very nicely identify alternative splicing within the androgen receptor in patients and actually predict resistance in patients in advance of therapy. So this is preliminary data, but it clearly works, the controls are working, and we think that this will be able to analyze patients, predict response to therapy, and hopefully the correct therapies to use. And what we want to do with this is identify therapeutic antibodies that bind these alternative spliced ARs and other proteins. Right? So this is generalizable to any protein made in the human proteome. So we can identify tumor-specific variants, and then we're very excited to be working with John Babcock at the CDRD to develop therapeutic antibodies. This timeline from antibody generation to deliverables about 2 years. So as part of this effort to identify some of the determinants of progressive disease, we've been sequencing matched primary and lymph node tumors. And this is an early example of that work. So we have the primary tumor here and the lymph node here, and we're plotting copy number. Right? So we're taking 100 million or so sequence reads, mapping them onto the genome, and plotting copy number. So these have pretty normal looking copy number here. And we see a little bit out here. Now if we look at these chromosomes here, in the primary versus the lymph node, we see there's quite a bit of copy number variation occurring here as these progress from the primary site to the metastatic site. Keep in mind this is like centimeters of distance, and yet we have this Darwinian selection driving these gains and losses. And so we're able to drill down into this and see that one of these is a gain on chromosome 7. So this is here, this is the lymph node here and the primary tumor here, and this is chromosome 7. We see this gain here, we see better here. Turns out that's EZH2, which is a transcription factor that's known to downregulate cadherins. So that might make sense, because cadherins are cell adhesion molecules, so suddenly we can move in a little bit easier. And we've confirmed this with CTAG and taken Huntsman by fish, and actually David Cox at the prostate center Michael Cox is working with Pfizer on developing therapies aimed at this molecule. This is another approach to finding sort of molecular determinants of progression. And this is where we work with Andrew McPherson and Cenk Salamat to identify fusion transports. So in this case, we're sequencing the genome and the transcriptome and looking for fusion events, so sort of like BCR-able events. And so we've had some success with this. This is an example of a fusion we found. This is PS3. We found this in our tumors. It turns out that this is both recurrent and androgen regulated. So it should be. Unfortunately, it was already published. That's okay because it validates the approach. Here's another case where we found a fusion between the TPD52 gene and this MRPS28 gene. Again, this is androgen regulated and recurrent. And this has already been published as well, TPD52, oddly enough by myself and Mark Rube. So this is where we showed that TPD52 at least is androgen regulated, and its copy number increases with progressive disease and prostate cancer. And so the challenge now is to show some biology behind this, and we're working on that. In each case, these are recurrent, and they seem to be restricted to prostate cancer and not seen in any normal tissue that we've looked at. So that's extremely interesting, and we're submitting a paper hopefully today on that. There's another thing we can do, which is look at sequence variants and ask if we can find mutations, for example, in genes like PTEN or RB that might cause progressive disease. Now, the odd thing in prostate cancer is that although PTEN and RB are deleted at high frequency, frequently heterozygous mutations, so one copy remains, nobody's finding mutations in these genes. So what I would call first-tier oncogenes escape mutation in prostate cancer. But sort of second, third-tier cancer genes seem to be hit frequently. And nobody really knows what's going on with this. I have some ideas if you'd like to talk with me later about it. But it's complex. So this shows some work where we've been doing with Stephen Jones, identifying sequence variants. We can't really call them mutations in the primary and the lymph node. In red are the sequence variants that are in common. They should be in common. They're related tumors. And I put a star here because it's the sequence here, MEG-F8, this is a gene. We find it a variant of this, maybe a mutation in our prostate tumors, where it shows up in 18 other kinds of cancer in the database search by Stephen Jones. So we're seeing this a lot. So these are variants, and these are in the primary and the lymph node, and these are the sort of frequency that are seen in other types of cancer at the database in Steve Jones search. So it's not really clear what this means. However, I think that we have an opportunity by integrating sequence variants with subnetwork analysis and alternative splicing to discover what some of these things might mean. And this is an attempt to do that. So this is integrating results from sequence variants in a number of paired primary and lymph node tumors. So alternative splicing, sequence variants, and subnetwork analysis. And then we've plotted in blue, I think, is the alternative splicing. I think red is subnetworks, and green is SNVs or something like that. In any event, we're able to identify the top networks and ask what they are. These are all of these are highly significant. So the first network or pathway that dropped out of this was axon guidance. And I thought, well, this is goofy, right? I work on prostate cancer, not neurobiology. But what we're doing here is we're saying, show us all the SMVs that are present in your lymph nodes, not in the primary cancer. So we're asking the difference and then doing a ingenuity pathway analysis. We get axon guidance. So I start reading about axon guidance. Well, what's a neuron have to do when it's growing? Chemotaxis, adhesion, migration, invasion, and survival. And it turns out that almost every protein in these pathways, the literature is split between metastatic cancer and neurobiology.